Okay, I realized yesterday after class that I probably should just make a recording of the main points I was going to make on Tuesday, and just make a recording, and that would have two purposes. One, our exam is on Thursday, and so there is new material for us to learn, and in chapter 19 and 20 still, and I thought I'd try to give it to you, as early as possible, so that you could, organize that and ask questions about it. Then also, I thought that would help us free up some time on Tuesday where I wasn't, pushing to continue to talk about stuff myself, but we could talk about questions, and we won't talk about new material on Tuesday, but we'll have plenty of time to go over the material, and so please bring lots of questions. So in class on Thursday, I went through these four items on this list. These are ways that scientists manage to help make early progress in understanding molecular mechanisms of cancer and help them identify genes that were oncogenes that could be misregulated or mutated and and contribute to cancer. And so we went through that. I just wanted to show you that again. Here's another way that comes through in chapter 20 about cancer biology. And so I want to talk through this. And so we've talked about this a lot. So the first one we've talked about a lot, uncontrolled cell proliferation. So cancer cells have characteristic properties that distinguish them from normal cells. And so this would be one. They're proliferating out of control and typically proliferating pretty fast. And I just want to show you that this list actually has 8 items we're going to get to. Okay, so the first one is this, and the book is adding to this in chapter 20, and it's adding to this big framework that we have. We understand this one very well. And I wanted to show you this because there are some other features of cancer that we haven't mentioned yet in our course. This is one we really dwelt on. And this list of 8 items doesn't include all cancers, but it's some of the main features in general of cancers. So then here's the other one that we're experts on, right? Failure to go apoptosis. So you see that list we're working on, and 2 of the items are ones we've said, these would be characteristics of cancer, is that they are undergoing uncontrolled proliferation, and then they are failing to go through apoptosis. Okay, all right. So our book talks about this next one, and it features a couple of these, and I'll just describe this here. And that is, if we take some cells, and this is just a dish, a sterile dish with some culture media in it, and we could take some normal cells and we can put them in there, and we can grow them, and as they grow we can separate them so they have plenty of room to grow, and they just keep growing, and plenty of nutrients and stuff like that. And those normal cells will divide, but eventually they'll stop dividing. The individual cells in our body are mortal in the same way that our entire body is mortal. And so there's a feature of normal cell biology that's seen in this. And there's a term for this, and it's called the Hayflick limit. And that just means there's a limited number of cells, divisions, that a cell from our body could undergo. If we take normal cells from a child, they go through many cell divisions before they stop. If we take cells from an old person, they go through a few cell divisions before they stop. So there's an aspect of aging in our normal cells that each cell is monitoring, or essentially counting. Okay, and so that's just that. And so these would be cell cultures And in the very first chapter of our book, the book talks some about cell culturing, and we just mentioned it briefly. But it's a pretty common technique to take some cells out and isolate them from a human or from any other organism and grow them in a dish. And if you're studying cell biology or molecular biology, lots of time you can ask very specific questions by growing the cells in a dish. Okay? Now, if we take and do the same thing with a cancer cell, and this is generally true of cancer cells, put that in a dish and grow it and separate it and keep it, under good conditions, it will divide forever. Cancer cells are said to be immortal in a dish. And so that's just a curious feature of cancer. And it's been known for years and years. And at first it was just a kind of a phenomenal logical observation that they have been somehow changed or transformed from a normal cell so that they are no longer mortal. Okay, so that's one of the features, is they'll just keep growing, unlimited growth like that. In the same context, there's another thing that comes up in our list, and it looks like this. For normal cells, if we put them in a dish, they tend to stick to the bottom of the dish, and they grow and they divide and they grow and they divide until they cover the whole bottom of the plate, and then they stop. And that's due to a natural, normal phenomenon that's called contact inhibition. And if we compare that to a cancer cell, cancer cells will grow until they cover the whole plate, and cancer cells lack contact inhibition. So when they fill the bottom of the plate up, they just keep dividing, and they start floating up in the liquid media and are not limited in that same way. So two features of cancer are that they are in a dish they're immortal, and they lack contact inhibition. Okay, so that's a cone here. And so that's here, unlimited replication, and that's what the scientists mean. It's not just that the cancer cells are growing fast, is that they'll just keep doing that forever if given the opportunity. And then here's this one, and this is the one that's really referring to that contact inhibition. Cell adhesion molecules are on the surface of cells, and we haven't mentioned them in our course, but they're on the surface of every cell, and it's really through those cell adhesion molecules touching each other that normal cells reach full coverage of the plate and then have contact inhibition. The contact inhibition, the contact between the cells is between these cell adhesion molecules. So that's this one, and we could reorder the list because we've got uncontrolled proliferation, then apoptosis. Then our next favorite thing to say is that they're immortal and they lack contact inhibition. Okay. Then the next one is listed here, and deal with it separately, but it really follows up on the first one, and I think that's why it's listed in this order here. And that is that cancer cells have less dependence on growth factors, and that's because cancer cells have mutations. So here's a growth factor like PDGF and the PDGF receptor, and then a pathway like RAS, RAF, MEK, ERK, ALK, right? So reduced dependence on growth factors because the cell has mutations in the signal transduction pathway. So here's the growth factor, and downstream of it, there are mutations. So that's why there's less dependence on the growth factor itself. Might not need it at all. Okay, so that's a pretty simple one that's really kind of tied to this one, is that you have uncontrolled proliferation, partly because you don't need the growth factor, because the growth factor pathways have mutations in them that make them independent of a growth factor. The growth factor is unnecessary. Here's one that is true of tumors, and it is a different story that we haven't learned about. So we'll just say here's a tumor, and these tumor cells are secreting molecules, including enzymes that degrade proteins. So these proteases are going to degrade proteins, and that really can get like two things from the, to advantage of the cancer. The one we really want to emphasize here is that the cancer cells are often said to be invasive. And so this is based on, these molecular stories, these molecular features of secreting these proteases. It results in a medical aspect of cancer, in that the cancer cells spread. They invade and they spread into other tissues. And so, just a brief one, but probably a feature you've heard of about cancers, and just what's the basis for that? And it is part based on secretion of these proteases. Okay, and then the next one, and we're already almost done with this, but just to follow through this, is they stimulate angiogenesis, and angiogenesis is a formation of blood vessels. And cancers, solid tumors, are a piece of new tissue, and they need to get blood vessels to grow into them in order to bring oxygen and to take away CO2, then also to bring in nutrients. And that's a feature of cancer, and it's a pretty simple and obvious feature. It's listed because for years scientists have been developing inhibitors of angiogenesis so that we could essentially starve and suffocate a tumor. And some of those are used as medicines to inhibit this angiogenesis, because cancer is usually found in adults, and in adults there's not much new blood vessel development because there's very little new tissue forming. And so if we just do a general inhibition of angiogenesis, we could block the growth of the tumor by blocking growth of these blood vessels, and we'd suffocate the tumor. We wouldn't get oxygen into the tumor, and we'd starve it. It wouldn't have good nutrients. The last one isn't true of all cancers, but it's a good example for us to talk about, and that is that the cells have abnormal differentiation. So it's a good one for us to talk about because it has some relevance for our stem cell biology that we're learning about, and also, that's messed up, sorry. I just pushed the wrong button. Okay. Well, sorry, got distracted by that. So the abnormal differentiation is this, and that when a cell normally develops and differentiates like a muscle cell, it stops dividing and permanently leaves the cell cycle. So these muscle cells no longer divide. That's why we have the satellite muscle stem cells, because we need new muscles. These differentiated muscles, our actual muscle fibers, they can only grow a very limited amount. And if we want bigger muscles, or we want heel muscles, we need to get new additional muscle cells. And that's pretty typical. Pretty typically, differentiation involves the rest of cell proliferation. Prior to that, the cells are going through the cell cycle. And so in some unusual cancer situations, the problem is really not in unlimited growth. The problem is the cells fail to undergo differentiation. And there are some blood cancers, some forms of leukemia, where the cells fail to differentiate. And in chapter 20, and this is just from the previous edition of the book, sorry. In chapter 20, it shows this. So here's a cell in the bone marrow, And we've talked about this, that there are stem cells in the bone marrow, and they're constantly replenishing our blood cells. And so here's a cell dividing, and it's going to go through differentiation to become a red blood cell. And there's a mutation where it doesn't differentiate. And as a result, these cells don't differentiate and they continue in the cell cycle. And we want to learn about that just because it provides a significant way to understand cancer of some cancer types that's different than uncontrolled proliferation or failure to go through procreative cell death. Here, the cell remains essentially as a precursor cell and continues to divide, and so that becomes a cancer because we're getting too many of these cells. Okay, and so that's our list of those 8 items, and we should be able to list some of those and explain some of those on our exam. So that's what I was hoping to get to in class on Thursday to kind of tuck in some early stuff with chapter 20. There's more to say about chapter 20, but those are 2 good pieces of chapter 20. Now I'd like to go back and finish up talking about chapter 9, stem cells, and then whatever remaining time we have, we can fill in more about chapter 20, because there's always more to say about that because of how it's pulling together all the different things we've learned in our course. So in chapter 19, the topics are programmed cell death and then stem cells. And this is how we look at stem cells. So adult stem cells, and we have our example of our satellite stem cells that are satellite muscle stem cells. And we want to be able to answer questions about that. Just mentioned that there are stem cells in the bone marrow. There's stem cells in our skin. There's stem cells in our digestive tube, so let's just say our intestine. And there's specific stem cells in our adult bodies that are dividing or ready to divide in order to maintain those tissues. And those are all collectively called adult stem cells. And so we refer to them as adult stem cells even though they're present in our bodies throughout our lives from a fetus, child, teen, adult. And we'll just talk about them and our favorite example. And so it goes back to these videos and the description of how the satellite stem cells could differentiate into muscles, and we could make any precursor cells differentiate into muscle if we misexpress MyoD in those cells, but we can't get a differentiated cell to differentiate into muscles. And so we want to go back to this because this is part of those chapter 19 concepts. And so here's the muscle fiber that's discussed in chapter 19, and these cells are differentiated, and in G0, the satellite stem cell is ready to divide when called upon to produce more muscle stem cells. And so that's a good example. One of the things we learned about muscles in the muscle videos is that when we express MyoD in an undifferentiated precursor cell, we can make it into a muscle. But if we misexpress it in a differentiated cell, we can't make a muscle. And one of the reasons we learned is because of DNA methylation. So in chapter 20, DNA methylation is talked about with regard to stem cells. Differentiated cells have quite a bit of DNA methylation. Stem cells have less DNA methylation than differentiated cells. They need to be ready to make a variety of cell types, and so they have less of this DNA methylation. They have some, and there's a lot of stories about that, but we're going to focus in our course on the idea that an undifferentiated cell has less DNA methylation than a differentiated cell. Okay, just that principle. Okay, and so the next thing to say is that when we're talking about adult stem cell, these are the options that stem cell has. So we can think of our favorite one, a satellite stem cell. Oops. Okay, and what that cell could do. So the first thing is that right now they're all paused in G0. They're waiting at the green light, and they're waiting until they're stimulated to go through the cell cycle. So that's one thing the cells can do, is just wait. And we have a variety of stem cells in our body that are waiting, and they can be stimulated to grow and divide. They're not differentiated, they're undifferentiated, so they can go through the cell cycle. They're just paused in G0. Okay, the second thing is they can go through start and divide. And when they divide, really there are two possibilities for stem cells. And one is the stem cell can divide symmetrically, evenly, to make two stem cells. And when we're doing that, we're expanding the number of stem cells. We're expanding the stem cell population, increasing the stem cell number. And that happens in a variety of settings, and to increase the cell population. Okay? Then the other possibility is when the cells divide, they can divide asymmetrically. And when they divide asymmetrically, they produce one stem cell and one precursor cell. That's no longer going to be a stem cell. So that's an asymmetric uneven division. This is a symmetric even division. Either we get two stem cells or one stem cell and one precursor cell. It's important to show here that when they divide asymmetrically, the cells are still said to be self-renewing. And that's really what the name stem cell comes from. Because there's a stem of cells that is growing like a stem on a branch, and it's producing these precursor cells over time. Okay? And the precursor cell, for example, in a muscle satellite stem cell, the precursor cell, that could divide some, and it varies, but it's going to divide and then eventually differentiate and become things like a muscle cell for the muscle satellite stem cells, or a blood cell for the stem cells that are in our bone marrow, like that. So these are the two possibilities. You go through and start and divide, and if you divide, you can divide symmetrically, expanding the stem cell population, or asymmetrically, and having a self-renewing stem cell population sustained while you're producing precursor cells to play a role. So that's 1, 2, 3. Then a stem cell itself can just directly differentiate. So the stem cell can just differentiate. And it can differentiate into a specific cell type. And that can be done by stimulating the cell to differentiate. So without dividing, it just differentiates into a final cell type. And then the last one, and it's an important one to recognize, is that stem cells are dangerous cells, because what if they grow out of control? And so stem cells, if they get in situations they're not comfortable with, they'll undergo PCD, and it's apoptosis. So they will decide internally to undergo apoptosis. And it's important to show this list because it's a way of understanding stem cells specifically. We hear a lot about them, and they can do a lot of different things, but their actual cellular responses are limited to this: either wait, or go and divide, either symmetrically or asymmetrically, or just differentiate into final cell type, or undergo programmed cell death. Okay? So, next we're going to talk about embryonic stem cells. And here are the features we want to know about embryonic stem cells, is that they are only in mammals. So humans have them, and all mammals have them. But say a fish doesn't. There's no such thing as an embryonic stem cell in a fish, or fruit fly, like that. Only in mammals. and only very early in pregnancy. Let's see if I have a slide of this. Yeah. So here's a mammalian pregnancy. So mammalians have internal fertilization, and the ovary in the female is going to release an egg, so there's be ovulation. And this shows in a human, the ovulated egg starts to migrate down the fallopian tube, and it's along this path where fertilization takes place. And so the egg cell has to be ovulated before fertilization, and then sometime during this path to the uterus, fertilization takes place. And when fertilization takes place, it's here in this image, so very early after ovulation in this image, the fertilized egg starts to divide. And so we have a 2-cell embryo, a 4-cell embryo, 8-cell embryo, and that's what these are referred to. And in humans, there's a varying length of time that's, we could say, about 1 week from fertilization to implantation. So when the embryo reaches the uterus, the embryo implants in the uterine wall and develops there. And embryonic stem cells are all the cells in the embryo. So the 2-cell stage, those are both embryonic stem cells, 4-cell. There are four embryonic stem cells, and so for this stage there are 100 embryonic stem cells. So all the cells in the embryo, and we'd say prior to implantation. Once the embryo implants, the cells start to become different, and we start to have them no longer be embryonic stem cells. So very early in the pregnancy, like in humans, about 1 week. So, no woman is ever aware she's pregnant when there are embryonic stem cells, and then they're done. They go on to become a variety of cell types. So embryonic stem cells only in mammals, only very early. They divide symmetrically, right? So stem cells have an option how to divide. These embryonic stem cells are dividing symmetrically. We're in an embryo, and they're just increasing the number of these cells in the embryo. Okay? And I think those are the features of embryonic stem cells that we want to mention up front, like this list. Only in mammals, only very early, for only about the first week in humans, pre-implantation embryo, and the cells are dividing symmetrically, increasing their population. Okay? So, for the embryonic stem cells, they have the capacity to form the whole organism. And that's shown here in examples, not from our book, about twins. And these would be identical twins. So identical twins are formed by splitting of an embryo, and they can split at different points in the pre-implantation embryo. When the cells are embryonic stem cells, the cells have the capacity to make the whole embryo, and they do. So if we split the embryo, we get 2 embryos. Okay? Embryonic stem cells have a low amount of DNA methylation because they can make every cell, as we just saw, and so they have low DNA methylation because they need the capacity to express any gene because they can become any cell type. So I guess we should add that as another feature to this list is low DNA methylation. Okay, so we have 4 features that we'd like to learn about for our course. Okay, all right, and so because of that, those characteristics, embryonic stem cells can be taken from an embryo and they can be grown in a dish like we talked about. So here's a sterile dish with liquid nutrients in it, and these are embryonic stem cells. And they're dividing symmetrically, so we can increase that population. And when they're dividing outside of the embryo and it doesn't implant, they'll expand a long time. There's a limit to their expansion potential, but they can divide a lot and produce a lot of embryonic stem cells. So that's one reason that people really like to study embryonic stem cells, is it's possible to get lots of them in a dish to study them. Okay? We could take these undifferentiated embryonic stem cells and we can get them to do things, right? So what can we get them to do? Well, they can do the things we get stem cells to do. One is they can just pause. We can just get them to wait in G0. Two is they can divide, and when they divide symmetrically. And three, well, I'm going to have a hard time remembering this. They can differentiate. And then the fourth one was they could go through apoptosis. So here we have these cells growing in a dish, and they're stimulated to grow and divide, and then we can change the conditions and get them to differentiate. And that's something that's done a lot by stem cell biologists, because stem cell biologists would like to take stem cells and get them to make new, fresh, healthy tissue to help somebody, say, for example, maybe somebody that had a heart attack, we could get them some new heart cells. Or maybe that somebody that has had an accident and injured their spine, we could get them some new nerve cells. Or somebody who's old and has lost some brain function, like due to a stroke or Alzheimer's, we could give them some new neurons, like that, okay? So a variety of different cell types. And so scientists are able to take embryonic stem cells and stimulate their differentiation in a dish to become any of a variety of cell types. And the way they do that is with these signaling factors that we've learned about in our course. Growth factors are being used to stimulate the cell to divide and grow here, and then the scientists simply switch the growth factors that are present in the dish to stimulate the cells to differentiate. It's a tricky job because it's not a very natural process, and so stem cell biologists often are frustrated because the cells they're trying to do experiments on will go through apoptosis because they're treating them in a somewhat unnatural way. But that's the logic of a lot of stem cell biology, is can we take these stem cells and get them to differentiate to contribute to the tissues in an organism, and tissues we need some help with, okay? So that's what's showing here, and it's just showing this idea that there are these factors that are just added to the dish, and these would be purified proteins that are added in kind of similar to how a person gives themselves injections of insulin. Insulin is a signaling factor, a signaling molecule. These would be a variety of different signaling molecules like we've learned about in our course, and scientists often refer to this as sort of a cocktail or a mixture of these signaling factors. And so we can stimulate the cell to grow and divide, and then in this case, if we just remove these factors, the cells will differentiate. And if we add specific factors, we can get them to differentiate down these different possible pathways. Okay, so that's the first round about embryonic stem cells, is to grow them in a dish and expand the population by symmetric cell division in a dish, then get the cells to differentiate to contribute some new cells. Okay, so when cells are differentiated in a dish, we can make some cell types like muscle and nerve and stuff like that, but the cells seem to lack guidance to really make complex tissues, like a really good muscle that would contract, or like something like, could we get the cells to differentiate to form a kidney? And the answer is no. Kidney has got too many complicated parts to it to really have it created just in a dish. several years ago, scientists found out that they could help cells in the following way. They could take a kidney, say, so this is just a kidney, and they could take a kidney and they could remove the cells. So they could kill the cells and remove them, and they call that decellularizing the kidney. And when you do that, if you remove the cells, gently and just with biochemical treatments rather than, grinding, we end up with a mesh of material with no cells. And this mesh is called the extracellular matrix. And it's a kind of extracellular framework that really helps hold the cells together. And part of it is structural. So the extracellular matrix is, for example, where the protein collagen is, and other proteins that are in cartilage. So, in your nose and so forth, you have a lot of cartilage, and that is actually extracellular matrix. And those are proteins and other polymers that were secreted by the cells to provide some strength and some support. So if we decellularize a kidney, we're left still with the framework for the kidney. And scientists discovered, again, several years ago, 15 to 20 years ago, if you take a decellularized kidney extracellular matrix and you add embryonic stem cells to that the material in the extracellular matrix is sufficient to guide these cells to make a new kidney. And that's exciting, but notice that we aren't getting more kidneys. So we're not able to grow a kidney for a patient and say, here's a new kidney, we have an extra kidney. We can just replace a kidney like that in a dish. But it provides some really significant information about how stem cells are guided in their differentiation process. Okay, so just a key thing about differentiation like that. And this is just from an intro bio textbook. Here's the cell membrane, like that. And outside of the cell, we have this extracellular matrix material, and includes collagen and other proteins like fibronectin, and then it includes other things like this long chain polymer of polysaccharide. And those include some polysaccharides that you may have heard of, because people take diet supplements who have issues with arthritis and aging, because in arthritis, it's the cushions in our joints that sometimes cause problem because they're getting kind of worn out and thin. And those cushions are made up of extracellular matrix cartilage material. So some of the proteins and polysaccharides in the extracellular matrix would be ones that you'd go like, oh, I've heard of that, because they are in diet supplements. All right, so that's the extracellular matrix. So the next thing is that scientists realize that what we need to get these stem cells to really form tissues that we want to form, we could use the extracellular matrix. So it's a question of getting some stem cells and the extracellular matrix. And so what some companies have started to do is they use 3D printers to print extracellular matrix mesh with stem cells in it. And so the ink in the 3D printer is composed of stem cells plus the different components of the extracellular matrix that they want to work with. And so companies are starting to do this, and just doing it in a really simple way now, but 3D printing technology is really dramatically taken off, and in the background includes 3D printing of living tissue. And so here's just kind of a visual example of that somebody printed this tissue, and obviously it's really simple tissue, so far, but there are big goals with it moving forward, okay? All right, so that's what we want to talk about with regard to embryonic stem cells. When we were talking about the embryonic stem cells, talked about the idea that some signaling factors could be added to help maintain the embryonic stem cells in a dish. And that's an example of what people call a stem cell niche. And a stem cell niche means the environment that the stem cell is in. And the best one to think about is in the bone marrow. So in the center of our bones, in the bone marrow, there are stem cells dividing right now, replenishing our blood cells. And so here's one of those stem cells, and it's growing and dividing, and in that case it's dividing asymmetrically, so it's maintaining a self-renewing stem cell population, and it's making precursor cells that are going to go on to form the blood. In the bone marrow, there are cells surrounding the stem cells, and those cells are secreting factors that help maintain the stem cell, help guide it. So the stem cell isn't making choices just on its own. Its neighbors are also helping it make choices. Helping it make choices of should it pause and wait, or should it go through start, or should it differentiate, or should it go through apoptosis. And so scientists have known that and known that these are signaling molecules, and so they'll take purified examples of those and use them as this cocktail in a dish to create a stem cell microenvironment. So embryonic stem cells have a microenvironment. It's the early embryo as it's moving down the fallopian tube toward the uterus. Adult stem cells have a microenvironment like the muscle satellite stem cells, and our muscles are surrounded by cells that are helping them be stem cells and helping them in their developmental roles. Okay. And so here's just a journal article where they're using this terminology of microenvironment, or again a niche. And a niche is a word from ecology, and it's where the organism lives. It's in it's in its environment. Okay, and so there's a lot known about that here along the blood vessels in the bone marrow, and different areas in the interior of the bone, and how this microenvironment is maintained, and how the cells are stimulated to grow and divide to form the blood precursor cells. And you can see it gets complicated with a bunch of different signaling pathways, signal transduction pathways and signals to help guide the ongoing development in that. Okay? And like I said, I just made a slide like this where scientists could use those purified factors to recreate that bone marrow microenvironment in a dish, just to maintain those stem cells in a dish and grow them and then study them. Okay, and our book talks about that in this chapter, because the idea of this puts together the idea of let's get some stem cells growing and then let's use them to make cells that the patient's body needs. Okay, so the last one on this list is induced pluripotent stem cells. So adult stem cells, embryonic stem cells, and pluripotent stem cells. And so these are normally naturally occurring, and induced pluripotent stem cells are the result of an experiment. So they are not naturally occurring. And about 10 years ago, some scientists won a Nobel Prize for understanding stem cell biology and creating stem cells in a dish. And this scientist did his work back in about 1960, and what he showed was some of the first examples of the concept of genomic equivalence. And it's important for us to remember that concept we have in our course, and that genomic equivalence is that every cell has all the genes. Okay, and then we've talked a little bit about this scientist's work in our course. This scientist's work was all done like from 2006 to 2012, and then after that, but his Nobel Prize work was done then. And what his idea was this: when we talk about a myoblast differentiated to a muscle, we could look at the gene expression, and we can look at it by studying the RNA. And when we look for the RNA of the myoblast, of the myogenin gene, just to go one example, when we analyzed, we would find no myogenin RNA in a myoblast, and we'd find myogenin RNA present in a muscle. So now imagine we just do that for every gene. And we would find several genes that were expressed in muscles, but not myoblasts. And these would be the genes that are important for making muscles. And so people often just call them generally muscle genes or muscle-specific genes. And all the different tissues in our body have tissue-specific sets of genes. So our eye cells express certain genes, our brain cells express certain genes, our muscle cells express certain genes. And we can analyze those by looking at the RNA levels in the different cell types. And if we looked at all the RNAs, we could find all of the muscle-specific genes and all the brain-specific genes and so forth, okay? So what Yamanaka realized is we could do this and analyze the RNAs, but what we could do is we can, instead of using a myoblast, we could use a stem cell and compare it to a precursor cell, right? So the stem cell divides and forms another stem cell and a precursor cell. And this precursor cell is not a stem cell. So how about we look at all the RNAs in a stem cell and a precursor cell, and we find those RNAs that are specific to a stem cell. And we could call those stem cell specific genes. What Yamanaka called them, and scientists call them, is they say they're stem cell identity genes. That's what gives the stem cell its identity. The logic is the same with the muscle. Muscle genes are kind of like muscle identity genes. They're expressed, and that's what makes this cell a muscle. It's also a little bit like circular reasoning. This is a muscle because it expresses muscle genes. These are muscle genes because they're expressed in a muscle. And that's a really powerful statement because it helps us understand the molecular differences between tissues and between cells. So we can identify these stem cell identity genes, and we don't need to learn the names of them, but this is what Yamanaka did with them. So he would take the DNA for a stem cell identity gene, and he would take the transcription unit that's going to code for the stem cell identity gene, and he would swap it with the regulatory gene of a gene that's expressed in a precursor cell. And then he would put this transgene into this cell, and that would lead to the expression of that stem cell identity gene here. And Yamanaka did that for several of the genes that they found were expressed in stem cells, and they found that if they expressed as few as 4 of the stem cell identity genes, they could turn this precursor cell or any other cell into a stem cell. So we take any cell, even a differentiated cell, and we could express or misexpress for these 4 stem cell identity genes, and this cell would become a stem cell. And this stem cell is called an induced pluripotent stem cell. And scientists are excited about that and have been studying that for the last 10 years because this brings up the possibility that we would take a patient's own cells from their body and misexpress the stem cell identity genes and get them to make a stem cell that is from their body. So perhaps we could grow a kidney in a dish and it would be the patient's cells. So there would be no risk of graft rejection because it's an autologous or the patient's own cells. And so that's a reason why scientists are studying these induced pluripotent stem cells. And the book talks just a little bit about the history of that work. This is Gail Martin, who is an early researcher in the field of growing cells in a dish and getting them to differentiate. Another one who was early in this field is, I think, the only molecular biologist in Maine that, really, or he wasn't a molecular biologist, but the only really biologist in Maine that would make it into an intro molecular biology textbook, and it's a guy named Leroy Stevens, who worked in Jackson Labs up near Bar Harbor. And just a picture of Leroy Stevens, and he studied mice, and he studied the development of some unusual cancer cells in mice that could behave like stem cells in forming a variety of different types of tissues, like that. So lots of interesting biology, past, present, and future in that field. This just shows something that might be confusing to students, and that is that these are the 4 genes, and again, you don't have to learn the names of the genes. One of them is c-Myc. There's one thing we're going to mention about them, and I'll mention about it in this slide, but we don't need to learn the names of those genes. It's these 4 genes, and to get the genes into the cell, this is a little bit confusing. The scientists will use a virus, because viruses can infect cells really efficiently, and they can infect every cell. The virus that's used is a defective virus that can't cause sickness. It has mutations that block its ability to cause any sickness or any trouble, and it's just used as like a package to get the DNA into the cell. Now, what do we want to know about these? Here's what we want to know about them, is that all of 4 of these are transcription factors. And that's not surprising, because transcription factors are going to activate several genes. And to be a stem cell, we want to activate several genes. Then the next thing to say about them is MYC will stimulate the cell cycle. One of the things, we know about the stem cells is they need to be ready to go through the cell cycle. Yamanaka could even take differentiated cells that were permanently stopped in G0, and he could get them to become induced pluripotent stem cells. And one of the reasons is because this expression of MYC will stimulate the cell cycle. Now, another reason to mention this is because we know MYC is a dangerous oncogene. This is the normal wild-type MYC, so it's not growing out of control, but it's dangerous to play around with MYC. And so none of these induced pluripotent stem cells have been used yet in human gene therapy, because scientists are still working with them and learning about how to control them and stuff like that. Then these other 3 are transcription factors, and they're involved in regulating DNA methylation. And that's satisfying for us because we know that differentiated cells are methylated and stem cells have less methylation. And so one of the things we're going to need to turn this into a stem cell is to remove some of those DNA, the methyl groups on the DNA. So Yamanaka's work is really satisfying because he found these 4 transcription factors that could activate a set of genes, express them, and stimulate the cell cycle, and also decrease the amount of DNA methylation on the DNA. And here these are, and I want to show you this because you don't have to look at this at all. You don't need to know the names of those, and you don't need to know what the book is trying to explain here. These 3 transcription factors have a kind of a complicated feature of positive and negative feedback on each other. So the book talks about it all, some here, and we don't need to dwell on that. Okay, so here's the book just describing that. Well, it's not, I guess, the book, but just going back to that image of now what we do is instead of having embryonic stem cells in a dish and maintaining that stem cell microenvironment and then changing it so that we can get differentiation, today scientists will start with maybe the patient's own differentiated cells. So this could be a skin cell, that's what they're describing here. They could misexpress the 4 stem cell identity genes that Yamanaka discovered to produce the induced pluripotent stem cells and maintain them in a dish, maintain a stem cell microenvironment in a dish, and then change that to get the cells to differentiate. So the induced pluripotent stem cells are replacing embryonic stem cells in research and in healthcare. Okay, so there's more we can say about chapter 19 and clarifying what we want to get out of it. But those are the most important things I wanted to say, so I'm glad I got to say them before Tuesday, so we can open up possibilities of answering questions about chapter 19 on Tuesday. There's a little bit more to say about chapter 20, and so I just want to show you this. And so we have that, the image that we've talked through, and that list of 8 features of cancer. The book is really divided into some general information about cancer, and then molecular mechanisms, and then therapy, treatment, like the CAR T-cell therapy, and I've mentioned this a couple times in class, or the tamoxifen. And so that we'll understand chapter 20 by plugging into therapy, what we already understand, and adding some more to it. And we'll understand molecular mechanisms based on the ones we already understand, like RAS, MYC, and so forth, but we'll add some to that. And then this part of the chapter we'll read through, and we want to learn it, but it's some general information about cancer, and we won't put really big emphasis on that part. There'll be, a small number of questions about that. Okay. So here's an example of the general stuff. The book says this, and it's just really satisfying to see this. It says the most cases of cancer are these 3 basic types of cancer. And they are solid tumors and blood tumors. So tumors of the blood aren't solid tumors, they're cells moving through the blood or the lymph system. And of these, the solid tumors are generally carcinomas or sarcomas. Carcinomas are almost all of human cancers. They're the most common ones. And they're malignancies of epithelial cells. So lung cancer and cervical cancer and gastrointestinal cancer, those are all epitheliums. Epithelia are sheets of cells in tissues. Then sarcomas are rare, and they're solid tumors of bone and muscle and cartilage, right? And so you may have heard of somebody who had bone cancer, but it's pretty rare compared to lung cancer, breast cancer, uterine cancer, and so forth. Okay, then leukemias and lymphomas are tumors in cells that form blood cells, and so they're not solid tumors, they're cells that float around in our bone marrow and in our blood. Okay, so it'd be good for us to learn this because it's just a dramatic and simple story, right? What are the three main types of cancer? And it's not, breast cancer, it's carcinoma, sarcoma, leukemia, lymphoma, and then, which is most common, like less common, like that. Yep. Okay, so good example of this to, learn from this and learn some of the basic knowledge from this. You don't need to learn the details like this. You don't need to learn the ranking of these specific cancer types or their prevalence. So, right, right, that's just, not the focus of our information in our course. There's some nice description here about the idea of a progression of a cancer. And so this shows this in this image, that the cancer starts out growing slowly and initially is a precancerous small patch or bump or lump, and then it starts to grow more. And we see some of the features of cancer, the 8 we listed earlier about uncontrolled proliferation and failure to go apoptosis and so forth. Some of those are represented here. And the cells grow, and they start to grow more, and then they start to spread and invade other tissues, like that. Here the book is talking about that contact inhibition, and that tumor cells lack that contact inhibition. They're also immortal, like that. We talked about this in class on Thursday, and our most important example of that is the HPV virus here, and it's a DNA virus, like that. So we learned about that. It's going to want to try to get that host cell that it infects to help it copy its DNA. Okay, and so here are just a couple slides I grabbed just to show you that in chapter 20 there's a lot of talk of genes. There's talk about the oncogenic retroviruses and how those are formed. So we've talked about oncogenic retroviruses for quite a while. Here are examples of them. And the book will use new oncogenes that we haven't learned about. Like here's an oncogene called ABL. And that's not going to be our big emphasis in learning in this chapter. We've talked about some new genes like Jun and Phos that come up in this chapter. And but we can deal with these on a case-by-case basis, and abelson is not, and we don't really need to know the specifics of abelson in our course. The book talks about other molecular aspects of this. This is talking about point mutations in RAS. So again, something we already know about that we learned, and originally was part of the first part of our course on the first exam, but we can ask those questions on the second exam too, because here the book is in chapter 20 bringing up these concepts. Great. Chromosomal translocations are dealt with. Here's that RAS, RAF, MEK, ERK, ALK pathway. So again, you don't have to go back to study the early parts specific for chapter 2, but here we're learning about Phos and expression of Phos. Okay, so you do need to be able to understand those pathways well enough to be able to know how they fit into the new material for this second exam. And we looked at this briefly also, that beta-catenin can also be functioning as an oncogene, because look what it can induce down here, and we talked about this earlier in our course in chapter 18. We can stimulate the expression of c-MEK and cyclin, like that. Okay, and there's some talk in chapter 20 about following up on chapter 19 and apoptosis, and so modulating apoptosis. Here's a pro-apoptotic regulatory factors being regulated by AKT. So we learned for our first exam, AKT regulates FOXO. AKT directly regulates the apoptosis and indirectly regulates apoptosis, like that. So that interplay of stimulating proliferation and thinking about programmed cell death. Okay, and I think I'm going to stop with this. And this just shows that earlier in our course we learned about PI3 kinase, that can be activated by a receptor tyrosine kinase receptor, and when it gets activated, it adds a phosphate to PIP2 to make it PIP3, and that's what activates AKT, and AKT can stimulate cell proliferation and survival. So that's what this enzyme does, and in chapter 20 it points out for these enzymes that do these growth responses on these growth response pathways, there are other proteins that will stimulate the opposite chemical reaction. So this P10 actually removes a phosphate from the PIP3 and takes it back to PIP2. And so this PI3 kinase has a little yellow mark around it because the book always tries to put look around that, where it's been shown that this is involved as an oncogene in stimulating cancer, and it's because it could be mutated and stimulate cancer by stimulating AKT in the absence of a ligand bound to the receptor tyrosine kinase. And so this is doing the opposite. So in a sense, P10, our book talks about this, P10 is a tumor suppressor because it acts in the opposite direction of activity of the PI3 kinase. So PI3 kinase could be considered an oncogene because when mutated, it could contribute to out-of-control stimulation of the cell cycle. P10 would be considered the opposite, or a tumor suppressor. So we could get a mutation in the PI3 kinase gene, and we could get out-of-control cell proliferation. We could get a mutation in P10, and that also could contribute to cancer because we're no longer shutting off this PI3 kinase pathway. Okay? And then there's some parts in chapter 20 that we just don't need to learn about, right? Because we skipped lots of chapters in our book, and here is following up some concept in a chapter that we didn't talk about. So when you see figures like this that look like a bunch of new information, mark that page and bring a question to class on Tuesday about what we need to cover. Okay? And I think I'm going to stop there with this CAR T cell therapy slide. Riley's going to come in one more time the final week of our class and just talk about the actual, patient treatment process. But we've learned a bunch from Riley, and there'll be a couple questions about on the test about what Riley has taught us. Okay, so I'm going to stop there. And again, I'm just trying to help us move forward. And we can just go through this and everything else on Tuesday, because we'll now have plenty of time to ask questions. Okay, so that's it. Thanks a lot.